morning, everyone. Thank you very much for this kind introduction. I would like to thank the organizer for giving me the opportunity to be here and explain the working that we are doing in this topic. Also, I need to apologize for sending a message that can be very scary, because plastics are everywhere, and we are really interested in the presence at the nano level, as you will see here. Well, maybe you remember this cover from National Geographic discussing the potentials of the pollution, the plastic pollution everywhere, by mimicking this iceberg and this plastic bag. And this is a picture on the Everest showing how the people is releasing a lot of plastics there. But when I started to perform experiments with the NxT for the first time, and Jolene Bradford asked me for providing a testimonial, I was really surprised about how the NxT can make visible the invisible. And that's the main reason, because we were really interested not in when looking at the plastic pollution, looking at the presence of these nanoplastic particles. The classical technology can have several limitations related to the particles that they can measure, but the NxT and nanocytometry can help to detect these very, very tiny and very small particles. The main reason is that, as usual, I'm not worried about the plastics, the microplastics that we are. Of course, I'm worried, but the real problem is related to the nanoplastic, the unseen problem, and we are continuously sending plastics to the ocean. The European community is trying to put a lot of money in this field to try to stop this continuous pollution of the seas, that at the end is also polluting all the ecosystems, and this should have an important impact, not only in human health, but also in many animals all over the world. Of course, we are moving at the nano particles. Just to show you these comparisons, during the pandemics, we were also measuring the coronavirus, and we were very successful identifying this tiny virus on the NxT. And this provides us a very nice approach to try to study nanoparticles using this powerful technology. As you can see here, we are moving at this scale, at the same size, near the same size of the coronavirus, with an estimated size of 70 to 120 nanometers. I hope this video will work. Again, you can see the massive plastic pollution on our oceans, which is really worrying. And only few people is doing research at the nanoplastic level. Then we decided to start a series of experiments in my lab. I think that this project started 2 years ago with preliminary experiments to adapt the technology that we have published very recently, as was shown yesterday. And this is the classical portals of entry defined for this entrance of plastic particles in the human body. One of them is through the skin by dermal contacts, like some toiletries and cosmetics can enter via the dermal contact. Inhalation has been proposed as a potential entry of these plastic particles, and almost all the people is really worried about the quantity of plastics that we can ingest daily. And as you will see here, there is an important estimation about the plastics that we can ingest weekly. And the main important topic is how this nanoplastic exposure can affect our metabolism. The problem with plastics is that not all are toxic, but they can attach very toxic molecules, and this can modify our metabolism. This can also be related to the absorption of nutrients with the energy metabolism, of course, the immune response, as you will see later. And some other people is trying to study the cytotoxicity of these plastic particles, performing in vitro experiments with nematodes and other single organisms. Sorry for say single organisms. And also this can be also affected. This nanoplastic exposure can also affect our behavior. This is really shocking because we are especially worried about the single plastic using. But as you can see here, 35% of the plastics that we are sending to the oceans is related to the washing machines that we are using daily. And the 28% of these plastics that we are sending to the ocean is related to the abrasion while driving. This is the tires that they are degraded. Also, the city dust in the industrial zones, the road markings, the boat coatings, and only unimportant, but when compared with the vast majority of plastics that we are sending to the ocean, is related to this single plastic using that we are daily manipulating. This is a very short, a very recent research performed in PubMed showing the search query showing the plastic pollution from the beginning from this year. As you can see here, there's an important number of publications because the scientific community is very sensitive and they are extremely investigating this topic of the plastic pollution. When you cross plastic pollution and microplastics, you can see here that this number is decreased, but also the people is investigating the plastic pollution and nanoplastics. And this is a topic that we would like to show you today and try to answer a lot of questions that were performed yesterday. Just to show you this very nice paper, because this research team was studying the nanoplastic transport to the remote high altitude Alps. Plastics are everywhere, are detected in the Everest, are detected in the Antarctica, and in many remote locations. And they decided to collect the snow in this observatory in central Austria. And after collecting many different samples, they were able to use a dispersion model, as you can see here, by supercomputing modeling, showing how we are sending all these plastics to the sea and basic on the climate change, how these plastic particles can migrate to the Alps and be accumulated during the winter and also accumulate in the snow. And then that's the main reason, because we are continuously sending these microplastics to the ocean, and then these plastics will be related to the changes in the global weather. And this is just to show you a different picture, because this is really common how we are having an important impact in all the ecosystems all over the world. And also we were also interested in measure the plastic accumulation, not only in humans but also in water. I'm in contact with people from the animal biology at the Faculty of Biology in Barcelona, and they will bring me water and ice from the Antarctica, and we will try to measure the nanoplastic accumulation in this kind of samples. We decided to use, as it was explained yesterday, detect this nanoplastic detection, this nanoplastics using the cutting instrumentation that we have, the NxT, because, as I was saying previously, current techniques such as the Raman spectroscopy can resolve only particles between 1 and 2 micron size, and this is insufficient to detect nanoplastic. And then we decided to use the Nile red. Nile red is a very well-known probe used to identify natural lipids. And then we decided to use this approach to study the plastics. The Nile red is quickly attached to plastic particles, and to perform this kind of experiments, we need to degrade all the organic matter and deplete, if not all, almost all the organic matter present in the human specimens, but also in other biological specimens that we are using. This is the method. We are always using the violet side scatter to discriminate small particles. And as you can see here, we are always using the H parameter, which is better to identify very small particles. In addition, we are using the specific filter kits for that are provided by Thermo to discriminate very small particles, and we are also using the violet side scatter kit to discriminate these very small particles. We are especially concerned about the potential contamination coming from airborne plastic particles, and that's the main reason, because we are always manipulating these samples under a BCL2 plus environment, which is a biosafety level 2 plus level, the same that we use it to identify the inactivated coronavirus. And then started by studying the presence of nanoplastics in peripheral anticoagulated peripheral blood. All the samples are always processed in triplicate, and we perform a quadruplicate when discordant, because we are using high-grade plastic, which is highly thermostable, and just to discard potential contamination coming from plastics, as one of the people and one of the people in the audience was asking. And when possible, we use also crystal tubes to avoid a potential cross-contamination originated in the plastic that we are using in our laboratory. And in addition, the Nile red is also titrated by staining blind microspheres, because we are moving to different lots, and we are performing daily many different experiments to analyze more and more specimens, and then we always stain blind microspheres just to be sure that we are able to detect the same amount of plastics varying from lot to lot when we change the Nile red stock solution. Just to show you the methodology, the basic methodology, this was done using the submicron beads from Banks Labs. You can see here the 200 nanometers, 500 and 800 nanometers here, and the nice separation using the violet side scatter, but also the very good separation, also using the blue side scatter. And then this is just to show you the presence of mainly nanoplastics. In here, we can identify the microplastic population, which is a very small fraction when we compare in absolute numbers the proportion of microplastics versus nanoplastics in the different samples that we have studied. Basically, we add 20 microliters of peripheral blood in potassium hydroxide 1%. And this is an old and classical method that can be used to degrade all the organic matter, and then we achieve a 1 ml final volume, and then we degrade the organic matter using this thermoblock at 60 degrees centigrade for 10 days. The original protocol is for 1 day, but or overnight, and we decided to extend the amount of time to be sure that we are degrading the most organic matter in the samples that we are analyzing. And this is just to show you electron micrographs that were performed when I was collaborating with the animal biology department at the Faculty of Biology in the Faculty of Biology, because we were really interested in the effect of pollutants on the eggshell. And we perform, this kind of digestion to remove all the inner cell membranes that are attached to the inner face of the eggshell. And we were monitoring the crystal deposition in this inner face of the eggshell. And as you can see here, you can easily distinguish the pores and also the pores, just to be sure that we are degrading effectively all the organic matter that is attached to the cell membrane, and just to confirm that we are trying to deplete the most of organic matter when we are digesting blood or other tissues, as you will see later. This is we daily perform highly exhaustive controls. This is the accepted noise that we can obtain from filtered and ionized water, and we will always detect this presence of nanoplastics, because the tubing is plastic, and the filters used are plastic too, and we will have a very good contamination. This is especially critical on Monday, because on Monday is the most difficult day too, because we need to run a lot of water just to start with the experiments, and we need to monitor this presence, because we are also always diluting our samples using deionized water, and that's the main reason, because we should control this continuously. And this is the classical stain that we can obtain in peripheral blood. And this pattern can vary when we compare different individuals and different specimens. This is just to show the digested and the undigested samples. And this is a true presence of nanoplastics. This signal is coming from lipid droplets that can be present in the undigested sample. And then these are the first preliminary results that we obtained using consecutive peripheral blood donors at the blood bank in my hospital. As you can see here, there is a threshold. This is the noise coming from, or the nanoplastic presence obtained from the ionized water. The noise coming from the potassium hydroxide solution that we use for the digestion of the organic matter, and trying to answer the question yesterday, as all the blood is collected in plastic tubes, we decided to dissolve and use water to monitor the potential contamination coming from the EDTA tubes, just to measure the noise and the potential contamination of this plastic, these plastic tubes that are used to collect the specimen. This is the results. This was done in triplicate, as you can see here, and this is a consecutive and differentiation accumulation of plastics in different individuals. And then we were really shocked about this distribution, and that's the main reason, because we are trying to develop a map of Barcelona, because we would like to obtain the blood of different donors, and using the postal code to try to obtain a map of the city for potential risk of exposure to nanoplastics. Then we started to measure, we continued these measurements. As you can see here, this is a summary of the healthy donors that we have analyzed in our lab. I would like to say that all the specimens that we have analyzed contain nanoplastics. These are newborns. These are patients with acute lymphoid leukemia, acute myeloid leukemia, chronic lymphocytic leukemia, multiple myeloma, lung cancer, idiopathic nephrotic syndrome, type 1 diabetes. And this is our result that we have obtained from mice, a question that was emerged yesterday, of course. And then I will explain later how we have to perform these experiments. And as you can see here, the newborns and mice are the less accumulating specimens in these experiments. And then we have also measured this variation related to the age, newborns from patient and donors from 1 to 9 years, from 10 to 19, 20 to 20 to 29, etc. And as you can see here, there is it seems that there is a progressive accumulation, which is non-significant, but at the end, there is a progressive decrease of a tendency in the decrease of the accumulation of plastics. But I think that this can be related to a metabolic process. Maybe these plastics can also be accumulated in adipose tissue. And these are experiments that we would like to perform in the future. As you can see here, we have compared the difference between sex and male and females have the same levels. There are non-significant differences, and the same for health versus pathology. And that was initially very shocking, because I expected, I don't know why, maybe increased levels in lung cancer or increased levels in autoimmune disease. And this is a really nice surprise, because we are especially finding a very different result that we expected originally. I don't know why this is. We started to measure other tissues and biological fluids, and as you can see here, this result is referred to this axis, and this bronchoalveolar lavage, the lymph node plastic accumulation, and the peripheral blood is related to this axis. But as you can see here, bronchoalveolar lavage have an increased number of nanoplastics, as you can see here. But we can also detect this presence of nanoplastics in cerebrospinal fluid, indicating that these nanoplastics are crossing all the barriers, as we demonstrated preliminary with the detection of these nanoplastics in newborns. This is related to the nanoplastic accumulation in disease for this format. I don't know what happened when I send your presentation. I'm usually using Keynote, and maybe because I'm using Mac, this PowerPoint is a little bit different when compared with the PC version. And now we are really interested in the accumulation of nanoplastics and how this plastic accumulation can be related to autoimmune disease. We have started measuring the accumulation in type 1 diabetes, and surprisingly, we have detected a lesser accumulation when compared with healthy donors, and also when comparing with a different population of pediatric patients which are suffering the idiopathic nephrotic syndrome. And we are currently investigating this plastic accumulation in type 1 diabetes, and we would like to extend this experiment shortly for multiple sclerosis patients. I don't know why and how this is a report from the World Wildlife Foundation that was explained in Singapore in May 2019, and explaining how much microplastics are we ingesting, and they estimate that the mass of microplastic that we can ingest is related to a credit card per week. And I was really shocked about this. This was on the news continuously, and was covered for many media. And then we decided to compare the accumulation of plastics in mice, and also compare these results with the accumulation in humans. The most important thing is that these mice are bred in ASPF environment. This is a pathogen-free environment using filter area, and they are drinking filtered water. They are eating irradiated food, but they are breathing a very good quality air. And as you can see here, there is this variation is significant. How the NOD mice and the C57 mice are accumulating less plastic when compared with humans, and this is, I think, that very worrying because it's, I know that the three different ways that we're exploring, this is the dermal contact, the ingestion, and the inhalation, and are potential ways for the accumulation of plastics. But this can indicate that this is non-suspected initially, but maybe the most common way to accumulate plastic in humans can be related with inhalation and breathing. No? And well, this is the $1 million question. What are the effects of microplastics in humans? I decided to perform this time-lapse video for 2 or 3 days. I don't remember. And adding, this is peripheral blood. And as you can see here, there is a macrophage trying to engulf a plastic particle. The glow makes that this plastic particle appears larger, and the size of these particles are only 3 micron size. And during 2 days or 3 days, the macrophage is trying to engulf this particle, and at the end, the macrophage will die. This indicates that the immune system is really busy in trying to eat these microplastics, but also trying to eat or to engulf these nanoplastics that are present in the blood. And this means that the immune system is doing a job different at the natural job that they should perform usually in the absence of microplastics and nanoplastics. Oops. And then just to have a global situation or more a more objective point of view that we are studying in my lab, we have started a collaboration with people at the Faculty of Biology, and then we started to collect fishes from the Balearic Islands, especially in this zone, which is a very well protected area. And just to show you the big quality of these waters. And again, we are not watching here microplastics. We are not watching here microplastics, but we started to measure the potential accumulation in water, but also the potential accumulation in fishes. And we have studied more than the 4 species that I'm showing here, but just to show you the most epipelagic, which are mainly in the coastal zone, and compare the different accumulation with the more pelagic species, and then measure the accumulation of plastics in this, and compare the different accumulation in these different species: Calamaris, Physis blenoides, Bulbus, and Lanthanitus crocodilus, which are a very common species that can have also an important role in the fishery industries. And this is just to show you the plastic accumulation when we compare the events of per milligram. 5 minutes? Okay, no worries. Because I will stop shortly. And just to show you the different plastic accumulation, when we go deep in the ocean, in the sea, we will detect the less accumulation of plastics. And when we are measuring the fishes in the coastal zone, we will have this increased amount of nanoplastics. And just to show you in this this current and ongoing research, how the plastic accumulation is growing over the years, as we have demonstrated in the different campaigns by collecting these fishes and measuring the accumulation of nanoplastics in muscle. Also to show you this comparison of the plastic accumulation over the world, and this tendency is similar when we compare this tendency in fishes. In conclusion, I would like to highlight that these nanoplastics can be detected in different tissues, and this is a really important message that they can cross biological barriers, such as in the case of brain accumulation, can enter the living organisms through inhalation, and as confirmed by its detection in bronchoalveolar lavages and in mice, the lower accumulation of nanoplastics could be related to the different air quality in facilities equipped with its HEPA filters, providing clearance of large particles. In fact, nanoplastics accumulating in human tissues overcome biological barriers with a significant fraction accumulated through inhalated air. And just to remember that we can save resources as a take-home message one. We love shopping online by using different companies. And can you imagine the amount of waste that we are continuously producing and also sending to the oceans? And this is meaning that we can save a lot of CO2 production during the process of this shopping online. And just to remember that the ocean is much more polluted than we think now. It's expected that in 2050, we will have more plastic than fishes in the ocean, and these are the plastic will permeate all the planet, and virtually nothing is known about the abundance of and distribution of nanoplastics in the earth. And I hope that this method that we have developed will contribute to better and to enlighten the role of nanoplastics in human health. Just to agree, acknowledge the very nice collaboration with Thermo Fisher for a long time, with Mike DeWarren and Jolene Bradford, also with Michael Zoe, that was previously in Life Technology and now is in Sartorius, and we obtaining these images using the IncuSite. The people at the Faculty of Biology, Jacob and Marie, that performed this very nice work in fishes, and the people at the Black Blood Bank, and people for providing pediatric samples, and of course the people in my laboratory collaborating in this specific topic. Thank you very much for your attention.